So the video is going to be broken up into maybe 3 or 4 smaller videos just so that I don't have to record anything really big and sadly find out that the recording quality wasn't very good. So here's the first of that series, and then we're just picking up where we left off. I think that was on Thursday. I think we started discussing the prospects of drug development where you might start with like 10,000 lead compounds, but by the time you go through the lead optimization studies and then preclinical trials and then clinical trials, usually you get down to just one drug that makes it to the market. And so before we start actually talking about the chemistry involved in the lead optimization and steps surrounding that, I think we want to talk a little bit about how the legal process actually uh, goes. And let's see, for the type of work that we do, your product discovery, synthesis, and then structure-activity relationship. That last one, structure-activity relationship studies, like the typical medicinal chemistry work that people actually do. But medicinal chemists like myself, the kind of work we would do sort of falls into these like early stage work. SAR typically is sort of the main bulk of the lead optimization efforts, and everything else like natural product discovery or synthesis, that tends to be sort of the phase before the lead optimization step. And so here's how sort of the once you have a lead identified, this is usually after natural product chemists and whatnot have identified a really good compound already. Um, this is how that process usually goes. So there's the discovery, and then of course all that study that happens in the discovery phase. This is still including like natural product discovery and maybe synthesis of it, just so you can do downstream SAR studies and stuff like that. If you do test the properties, bioactivities of the compound, those are all in vitro, meaning not in an actual animal or living uh, animals. People, these are terms that you hear a lot in this type of like testing, bioactivity testing context, so you might want to be aware of it. In vitro just means you're not using any live animal. In vivo, as opposed to that, usually means you do that the bioactivity testing in an actual animal, or sometimes it could mean a human, so like higher organisms. In vitro could mean either you're just testing against inanimate proteins and enzyme types of targets, or you could be testing your compound or compounds against living cells, but not like a multicellular organisms. Okay. And then typically the preclinical trials involve determining things like toxicity, pharmacokinetics, and pharmacodynamics in animals, like at least mice. A lot of times, this would involve little higher animals, like as a dog pet owner, I hate to say this, but like dogs or monkeys, that kind of things. And then sometimes it only goes up to mice, but mice almost always are sort of go-to animals for these things. And then once the clinical trial begins, of course, that testing is with people. And the progression of the drug development goes like this. Of course, somebody like a medicinal chemist, not a physician, contrary to maybe what the public thinks. MD people usually are not involved in almost anything except for the clinical trial, the very last step, just to make sure it works with people. But some scientists would identify a disease, and usually these days, uh, funding agencies would want you to sort of distill the pathology down to a protein or a single target, or maybe a multiple targets, but some sort of molecular targets. And then once you have those macromolecule targets like a protein receptor, something like that, you might be able to identify a lead compound through, I think last week we talked about random screening and stuff like that, right? And then um, after that, once you have a solid lead, and hopefully it passes things like Lipinski's Rule 5, but if it doesn't, that would indicate that maybe the pharmacodynamics is good because in your identifying of your lead compound, supposedly you did some in vitro pharmacodynamic activity testing, bioactivity testing, and then it was good. It was potent enough, and that's why you're excited about it as a lead compound. But the pharmacokinetic properties, maybe because the solubility is poor or there's a very oxidizable functional group that you know is going to be oxidized and metabolized by cytochrome P450 and stuff like that. So if you suspect the pharmacokinetic issues, ADME issues, right, absorption, distribution, metabolism, and excretion type of issues, then that's where the sort of the lead optimization step comes in. Sometimes, even if you have a good lead that's potent enough, you might still be concerned about the pharmacodynamics in that, like, maybe it doesn't give you a good enough therapeutic window, and therefore you want to increase its potency and efficacy. So you might be trying to optimize both pharmacokinetic and dynamic parameters, but the lead optimization is you change a little bit about the structure of that small molecule, and that almost inevitably involves some sort of structure activity relationship, like we talked about it. And then once you land on a few good optimized compounds that are optimized further from the lead compound, then you might do some preclinical trials. This is where you would do animal testing to get some more realistic than in vitro type of toxicity assays. Usually, even at the in vitro stage, you do toxicity assays against just sort of regular human cells or something like that, just so that you have some ideas. But it's hard to predict the organismal level toxicity. So this is where the whole animal study goes in. And sorry, I don't know how to move this camera window, so maybe you can't see very well. But then you go into clinical trials. There's three phases of them, sort of. Some would argue there's kind of a phase 0 as well as phase 4, but we'll talk about that. Then once it passes those, then an FDA gives the stamp of approval, the drug can move to the market. And of course, that process is kind of nebulous and abstract. So like I just wanted to show you like one story, one example, and that's this anti-cancer drug called midostaurin, which went into market in 2017. But its story really began in 1977, and hopefully this kind of gives you some idea about just how long drug development process normally is. Okay, and while the compound was isolated in 1977, the structure it was complicated enough that wasn't quite established until 1994. That was further confirmed by the first total synthesis, meaning somebody was able to make this compound from scratch from commercially available simple compounds in 1995, and then that's when Novartis, a fairly major pharmaceutical company, took over the development. Usually around this time, at this stage of drug development, big pharma companies has to get involved, or usually there's not enough funding just with like academic research grants and whatnot. What they did is, of course, lead modification, and I will talk about this in a tiny bit more detail, but I want you to notice that the compound that they were pursuing at this point after the lead modification work that they did is a little bit different than the parent natural product. And so they settled on not the parent compound, which is called staurosporine, but this derivative, midostaurin, in 2002, and the clinical trials went on for 15 years, which may sound like a long time. I would say this is slightly longer than average, but it's not unusual. And finally, the FDA approval was given in 2017. As far as the lead optimization bit goes, of course, that begins with the first total synthesis because you need to have a well-established synthetic route to the scaffold. Otherwise, you won't be able to make any analogs or modify the structure. So that's the starting point. But one thing they noticed is that the parent compound staurosporine, as promising as the pharmacodynamic properties were, um, for the most part, it had some major side effects. I believe that was discovered in the animal study. Like it had a neurodegenerative side effects, and then I think, if I remember right, there was some side effect issues with the liver as well. And during the lead optimization or modification, the goal here was to sort of, as much as they could, eliminate the side effects, and that was achieved when they added the amide functional group at this free amine to create myostaurin, and then that's what they proceeded with. But usually, there's a lot of research effort by people like our lab for the preliminary chemically oriented investigation that happens. So the early stage work is often can be adequately, to some extent, funded by sort of academic research grants. But when you get to the lead modification stage, that's when things kind of start getting expensive, and to make it worse, like academically, maybe not as interesting perhaps. And so this is often called the valley of death in the pharma business world, because most promising drug leads just get abandoned around this step. And so actually, in the past, and my PhD advisor, we started a company to address this challenge, but that wasn't quite successful. It's really hard to attract funding at the lead modification optimization stage. Once you can get to the clinical trial stage, right, then uh, you know after after that is you pass all the preclinical testing and mostly in animals and stuff like that after optimizing the lead, then it's not that difficult, perhaps, to attract investment and funding. But this is the hard part. And then, uh, yeah, so that's something you want to be aware of. And then if you remember that funnel diagram for the drug development, right? Like that, this is the part that sort of the funnel gets narrower and narrower, and that's part of the reason. Anyway, once you do make it to the clinical trials, because you file this thing called IND application, investigational new drug application with FDA, you go through phase. Like I said, some companies, some situations call for a phase 0 safety testing, but that's kind of outside the scope of our class. Um, the phase 1 clinical trial usually only involves something like a dozens of people. And typically, this calls for healthy volunteers, believe it or not somebody who is actually sick with the disease that the drug is intended to cure. And the reason is, at this early stage, we just want to know how safe the compound is. So we just want to see if the material, the drug, can be tolerated by a healthy individual, maybe like 20 people. It's pretty typical, and that's the focus, is safety. And then in phase 2, you scale up a bit. You would have not just dozens, but like usually hundreds of patients. And this time, usually you actually look for patients, volunteers, meaning like people who are actually sick. And one of the things that you look for and the focus on in phase 2 is the efficacy. And the reason is because placebo effect is really real and strong, especially when you think about it, like the patients that would volunteer for phase 2 clinical trial usually tend to be pretty like desperate, unfortunately. And so, of course, they would like to believe that something like this works, and so placebo effect is true and real. And so you look for an actual true efficacy in phase 2. By the time you sort of pass that phase, and then you get to phase 3, you take a little bit more time to do oftentimes what we call double-blind clinical trials, where both the administrating physicians as well as the patients don't know whether it has placebo, whether the drug is placebo or a real drug being tested or not. And again, you look more into efficacy in a more scientific manner because it's double-blind um, clinical trial without supposedly too much any bias, and then you also look for a little bit more long-term side effects because this phase is supposed to typically last a little bit longer than phase 1 or 2. And then once the drug passes this, that it shows that it's efficacious enough that the risk of small side effects is tolerated uh, by most physicians' judgment, and the side effects are not too bad, right? Then it passes. In reality, in the last so many decades, I think this is actually very prudent on the part of FDA. They sort of put out what they would call phase 4 clinical trial. After phase 3, the drug is technically in the market. Any physician can prescribe it to their patients. But once the drug hits the market, FDA still monitors it for so many years to look for potential side effects that were not caught by the population of volunteer patients in phase 3. Um, and the idea is that if there's really disturbing side effects that are found in phase 4 after it hits the market, FDA is supposed to pull the plug, and sometimes that happens. But anyway, that's the different phases of clinical trials. And with this in mind, right, like in the next few videos, we want to look at the chemistry involved in the designing of drugs, as well as the chemistry involved in the lead optimization processes. And then that would conclude our semester. So anyway, I'll see you guys in the next videos. Thanks.